Cellular Respiration by Cailyn Moriah. ATP synthesis occurs in both the mitochondria and the cytoplasm of the cell.
The overall equation is glucose combines with 6 oxygen to produce 6 carbon dioxide, 6 water molecules, and 38 ATP. There are 4 stages in aerobic cellular respiration, the first step being glycolysis. Stage 1, glycolysis. Glycolysis is comprised of an ATP load stage and an ATP payoff stage, both of which consist of 5 steps each. Glycolysis begins with the glucose molecule, and this is where the first ATP molecule is loaned. One of the phosphates off the loaned ATP molecule reacts with the glucose to form glucose 6-phosphate and an ADP molecule. The glucose 6-phosphate molecule will rearrange to form fructose 6-phosphate. At this point, another ATP molecule is loaned to this reaction. In this reaction, the phosphate from the loaned ATP connects to the first carbon on the fructose 6-phosphate to form fructose 1,6-bisphosphate and another ADP molecule.
The backbone of the newly formed fructose 1,6-bisphosphate splits into a glyceraldehyde 3-phosphate, or G3P, and a dihydroxyacetone phosphate, or DHAP, DHAP for short. DHAP then rearranges to form another G3P molecule. This total reaction results in 2 3-carbon backbone G3P molecules. Glycolysis now moves into the payoff stage, where it will return the 2 ATP that was previously loaned to the reaction.
Glycolysis payoff stage. First, G3P each give up 1 hydrogen and 2 electrons to NAD+, forming NADH. At the same time, each G3P combines with an inorganic phosphate. This reaction produces 1,3-phosphoglycerate, or 1,3-BPG. Both 1,3-BPG, being as reactive as they are, readily give up their phosphate to an ADP, forming ATP. After this, 1,3-BPG becomes 3-PG. Then, the 3-PG molecule will rearrange to form 2-PG. After its rearrangement, 2-PG will give up a hydrogen and a hydroxide ion, forming a water molecule. After the removal of water, 2-PG becomes phosphophenylpyruvate, or PEP, PEP. This molecule, PEP, is very unstable and breaks down, releasing enough energy to transfer one phosphate group to ADP, forming another molecule of ATP. At this point, the original glucose molecule has been broken down to 2 pyruvate molecules, each with a 3-carbon backbone.
Final results. There are now 2 NADH molecules, a net of 2 ATP molecules, and 2 3-carbon backbone pyruvate molecules. Stage 2. Pyruvate oxidation. After glycolysis, the pyruvate oxidation stage will begin. But first, the pyruvate molecules are required to pass through both the outer and inner mitochondrial membrane into the matrix. To do this, the pyruvate molecule will easily pass through the porous outer membrane. After it passes through this membrane, the pyruvate molecule will pass through the inner membrane with the assistance of a membrane carrier.
Once inside the matrix of the mitochondria, a coenzyme A, or CoA, attaches to the acetyl group of the pyruvate molecule, enabling the molecule to be oxidized while reducing NAD+ to NADH, thus removing the hydrogen ion from the carboxylic acid group. Then the remaining carboxyl group is removed from the molecule as waste in the form of carbon dioxide. Overall, this reaction results in a product of acetyl-CoA. Acetyl-CoA will now continue into the Krebs cycle. Stage 3, the Krebs cycle. We are now moving on to the third stage of cellular respiration, the Krebs cycle. The Krebs cycle is comprised of 8 catalyzed reactions occurring in the matrix of the mitochondria. The cycle begins with the reaction consisting of the acetyl-CoA from the previous reaction and the oxaloacetate that is pre-existent in the matrix. Through this reaction, citrate is formed and CoA is removed from the molecule. Citrate then will undergo isomerization and become isocitrate. The third step in this cycle occurs through a redox reaction when isocitrate is oxidized and becomes alpha-ketoglutarate. Then, with the hydrogen freed from this oxidation, an NAD+ is reduced to NADH. From here, alpha-ketoglutarate then binds with CoA and is oxidized by an NAD+, resulting in succinyl-CoA and NADH. All the while, a carbon dioxide is removed from the molecule as waste. The succinyl-CoA molecule now loses its CoA and becomes succinate through a reaction where an organic phosphate molecule from the matrix will replace the CoA group from the molecule. After this reaction, guanosine diphosphate, GDP, is phosphorylated and becomes guanosine triphosphate, GTP. Then the phosphate is transferred to an ADP molecule forming ATP. From here, an FAD ion, also pre-existent in the matrix, oxidizes succinate to form another coenzyme, FADH2, resulting in succinate forming fumarate. A water is then added to the fumarate to form malate.
End results for Krebs cycle: 6 NADH molecules, 2 ATP molecules, and 2 FADH2 molecules. Remember that this is the outcome for only one of the 2 pyruvate molecules. In order for the Krebs cycle to be considered complete, both pyruvate molecules must go through, thus yielding 12 NADH molecules, 4 ATP molecules, and 4 FADH2 molecules. Stage 4, electron transport chain. This is the final stage of cellular respiration, and it is known as the electron transport chain, or ETC. But first, a few important terms. Chemiosmosis, otherwise known as a proton pump. This is defined as the movement of ions from areas of high concentration to areas of low concentration through a membrane protein. This results in a release of hydrogen ions, which are later used to make ATP. The second important definition to know is mobile electron carrier. These are carriers that carry electrons between proteins in the inner mitochondrial membrane. The third and final important definition to know is electron carriers. These are substances such as ubiquinone, or UQ, and cytochrome C, or Cyte C, which carry electrons they receive through the oxidation of glucose. Okay, so the ETC occurs between the inner and outer mitochondrial membrane, and the purpose of ETC is to produce ATP through oxidative phosphorylation. ETC is comprised of 5 major complexes. Complex 1, NADH dehydrogenase. This complex is responsible for removing the hydrogen ions from NADH, forming NADH2. Complex 2, succinate dehydrogenase. This complex is responsible for removing the hydrogen ions from FADH2 and will be avoided by the hydrogen ions received from the complex 1. Complex 3, cytochrome BC1 complex. This complex is simply responsible for retrieving loose hydrogen ions from the inner mitochondria, such as the matrix, and pushing it in between the inner and the outer mitochondrial membrane. Complex 4, cytochrome oxidase. It has the same responsibility as complex 3. Beginning first with complex 1. At complex 1, NADH gives up its hydrogen atom and forms NADH positive. From here, the hydrogen is split into its positive ion and its electron is removed. The proton is pumped into the intermembrane space, and the electron is pushed through the inner membrane. The mobile electron carrier, UQ, carries the electrons from complex 1, NADH hydrogenase, to complex 3, cytochrome B, C1, ultimately passing over complex 2. The mobile electron carrier, Cyte C, then carries the electron from complex 3 to complex 4, cytochrome oxidase. Returning now to complex 2. Here, FADH2 gives up its hydrogen atom, becoming FADH. The hydrogen atom then undergoes the same situation that the hydrogen atom in complex 1 did. The proton from this hydrogen is then pushed into intermembrane space, and this electron will also be pushed through the intermembrane, as the electron from complex 1 did. However, rather than both mobile electron carriers, UQ and site C, carrying the electron from complex to complex, it only comes in contact with site C, which carries it from complex 2 to complex 3, and then from complex 3 to complex 4, and so on. Through these reactions, ETC has created an electrochemical gradient to power ATP synthesis. Chemiosmosis now begins. Hydrogen ions are now forced through a special enzyme called ATP synthase, which is embedded in the inner membrane. These hydrogen ions flow from the intermembrane space into the matrix. This flow of hydrogen protons provide energy to make the bond between ADP and inorganic phosphate. This is how ATP is made via oxidative phosphorylation. End results of ETC. Firstly, please know that one NADH molecule produces 3 ATP, and each molecule of FADH2 produces 2 ATP. So, with that said, in stage 1, 6 ATP produced, stage 2, also 6 ATP were produced, and then finally in stage 3, 22 ATP were produced. The final results of aerobic cellular respiration is 38 ATP.
Plus CO2 and H2O is waste. La 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 la.
Cellular respiration is now complete. Yay!